All right. In this part, in this screencast, we're going to be looking at essential questions defining osmosis, including hypertonic, hypotonic, and isotonic solutions. I think you've seen this before, but so it should be a nice review. You also need to be able to track how water will move across membranes, depending on the solution that a cell would be bathed in. And then lastly, we're just going to touch upon why it's important for organs to be put into a similar osmotic solution as the cytoplasm of the cells that make up that tissue. All right, so let's look at osmosis. Osmosis is defined as the movement of water across a semi-permeable membrane, okay? And it diffuses through that membrane, mostly through facilitated diffusion. But actually, there are some cases that you'll read about some molecules of water. If the phospholipids have a big enough kink in it, some particles of water can actually go directly through the phospholipid bilayer. But for the most part, they're going to travel through a form of facilitated diffusion through proteins called aquaporins. Okay, so if we were to go back and look at this, if these were water molecules here, to get from high to low water concentration per se, then they would probably go through a channel called an aquaporin. Although, as I said, occasionally they have been known to go through membrane, the phospholipid bilayer as well, being as small as they are. Okay, so here's what happens. Now, osmosis, in the context that we're going to be looking at it, tends to travel based on, like the water is going to move based on if there is a differential, if there's a difference between either side of the membrane. Okay, so if here's a membrane, right? Pretend that's a cell membrane. It's selectively permeable. Now, the water isn't going to freely, the water can go through, but the solutes in here, let's say, it cannot move through that membrane. So here we've got a difference in solute concentration. If you remember, solute is what is dissolved into the water, and the solvent in this case is water. So the water can move back and forth, but these solute particles, whether it's salt or sugar in this case, are in a higher concentration here versus here. Since the sugar molecules can't pass through the membrane, then the water molecules will actually move to try to even out those concentrations. So you can see in the end, the water molecules moved, and now it looks like it's more evenly concentrated. So that sugar solute in there is of about the same concentration on both sides. Okay, so it's going to move along. The water is actually moving along its concentration gradient because the water is more concentrated over here than it is here. It's less concentrated here because there's a lot more particles getting in the way. But as far as the solution, the solvent is concerned, it's going to move from low solvent to high solvent concentration, okay? And there's special terms that we use to talk about that: hypotonic, hypertonic, and isotonic. So if you put a cell into a solution that has a concentration of solute that is less than what's inside the cell. So outside has a low concentration, inside the cell has a high concentration. We use the word hypo and hypertonic as simply a comparative term. The cell, in this case, is hypertonic to the solution that it's sitting in. It's sitting in a hypotonic solution, meaning it's less concentrated with solute. So hypertonic cell, hypotonic solution. In this example, we have a hypotonic cell to a hypertonic solution. And in this case, it's an isotonic solution because the concentrations are even to each other. So what happens when you have a differential osmotic potential like this? So when the cell is in a solution that has a greater solute concentration or less of a solute concentration, then we're going to see the movement of water into or out of the cell. In this case, if the cell is in a hypotonic solution, the water is going to mostly be leaving the cell. Or sorry, take it back. It's going to be going into the cell. So here we have lots of water, not very much solute, hypotonic solution, and the water is going to move into the cell. So the cell should essentially gain mass. In this solution, this is a hypertonic solution where the cell itself is hypotonic. So The water in this case is going to leave the cell. It's going from hypotonic to hypertonic, okay? And then in this case, we're going to have water coming in and water going out, but there's going to be no net gain. So you can see the cell here has gained mass. The cell here has lost mass because it's lost water to the solution. And then here, the cell has both gained and lost, but there's no net change. So there's no net change in mass of this cell. Why this is important is if you put cells into solutions that are not isotonic, then things can happen. Like animal cells, they can actually gain so much water they could burst or lyse. If you put them in a hypertonic solution, they could lose so much water that they shrivel up and lose their function. Plant cells actually prefer to be in hypotonic solutions because they are not going to burst because they can fill up with as much water until they hit the cell wall. We call this a turgid cell. That's how plants like to be. It makes them happy. If they're in an isotonic solution, they actually get a little bit wilty and the leaves won't stand up. And if they're in a hypertonic solution, you'll actually start to see them wilt and the cell membrane will pull away from the wall, and we call that plasmolysis. If we are doing like a tissue or organ transplant, when we take that tissue or organ, in lieu of it has to be transported from one patient to the other, they have to put it into an isotonic solution. This is important because you don't want the tissue to lose or gain too much water in the transport. Okay, so now, the thing that's most important for you right now is that if you can talk about, if you could label the different solutions as hypertonic or hypotonic, and if you can then talk about which way will the water move based on that differential, okay? So hypotonic solution outside the cell is less concentrated, whereas inside the cell is more concentrated. Water moves into the cell. In a hypertonic solution, where the cell is hypotonic, the water will move out of the cell and it will lose mass. In an isotonic solution, you'll have a net movement, so there'll be no gain or loss in mass.